Hello everyone and welcome to AP Bio Unit 3 Review. This is episode 2 for Cellular Energetics. This chapter is solely focused on how enzymatic processes and metabolism occur. And this specific video is extremely concerned with cellular respiration and photosynthesis, and basically how the mitochondria and chloroplast work. So without further ado, let's begin this chapter because this is one of the hardest chapters of the semester. After this, things tend to die out and dim down. The content isn't as intense, but this chapter requires a lot of understanding of chemistry and a lot of understanding of biology together. It's not as intuitive. So let's begin. What kind of cells undergo cellular respiration? Well, all cells undergo cellular respiration. Remember, cellular respiration, and this is going to answer question 2, occurs within the mitochondria. And remember that the mitochondria originate from prokaryotes based off the endosymbiote theory, which basically said that a mitochondria was, and then ignore all this stuff on the end, it was from a previous recording, it assumes that the mitochondria was from a prokaryote that was engulfed by a eukaryotic or a larger cell and formed a eukaryotic cell. But yeah, if mitochondria are from prokaryotes, then prokaryotes can also undergo cellular respiration. The important part that you really have to recognize, though, is that those processes that occur within prokaryotes and eukaryotes for cellular respiration are different. Okay, where does this process take place? Two parts. Glycolysis takes place, and by the way, for the purposes of this video, assume that I'm going to be talking about eukaryotic cells because that's what the AP test is concerned about for this chapter. Where does cellular respiration take place? Well, cellular respiration takes place really in two big parts: the mitochondria or the cytoplasm. Cytoplasm is only for glycolysis, and some scientists even debate that glycolysis isn't part of cellular respiration, but the majority of it takes place within the mitochondria. What are the different parts of this organelle? Okay, so the different parts of this organelle are the following, and I kind of have this little diagram here for you guys to kind of look at. There's really four big parts that you have to recognize, and one of them isn't even significant to the content of this chapter. Number one is the outer membrane. This is just basically there for the content or for the purpose of this chapter. It's not important. But what is important is the inner membrane. The inner membrane is this region that I'm kind of just drawing in right now. Just kind of look at my cursor. It's between the inner membrane, aka the cristae, and the outer membrane. This is significant because one of the reactions that happens in cellular respiration occurs here. But yeah, that's the inner membrane. Then you have this thing called the cristae, which is the inner membrane. Now recognize the difference: inner and inter. Inter is the one between outer and inner. Inner is the cristae, the part that I'm highlighting or outlining right now. And lastly, you have the mitochondrial matrix, which is basically like this jelly-like gooey fluid that a lot of stuff happen in and is pretty important to the content of this chapter. So, what are the steps of glycolysis? So the steps of not glycolysis, my apologies, cellular respiration. There are really three steps, but I'm going to jump in and put glycolysis within it. There is glycolysis, pyruvate oxidation, also known as the link reaction on this study guide. Then there is the citric acid cycle, also known as the Kreb cycle. They're interchangeable. Know both of them because they may pop up on the AP test as one or the other. And lastly, oxidative phosphorylation, which goes hand in hand with the electron transport chain, which I will refer to as ETC from now on. So, what happens during glycolysis? So recognize the first thing, that cellular respiration, cellular rep, is a catabolic process. And what that means, if you watched my first video, is that it is an exothermic process that you break things down in. Okay? Now, what's being broken down in this case? Sugar, mainly. Okay? Sugar is being broken down. Glucose in particular, and the equation for glucose is C6H12O6. And what's going to be happening to sugar in glycolysis is that the sugar enters the cytoplasm, and that is where glycolysis takes place. Sugar enters the cytoplasm, and a bunch of enzymes basically come towards that sugar and start breaking it down. But one thing that you should really recognize is the stuff that is made out of this entire process, and that would be 2 ATP, 2 pyruvate molecules, pyruvate, and 2 NADH. Now I'm going to jump a little bit ahead in the review guide and explain what NADH is and eventually what FADH2 is going to be. That's not going to really happen in glycolysis, but NADH is made. And NADH is basically a derivative or something that originates from this molecule called NAD+. It is a vitamin, okay? Now, NAD+ is this molecule that typically wants to accept electrons. Why is that? Well, recall that an electron is a negatively charged ion. Well, it's not an ion, but rather a negatively charged subatomic particle. And NAD plus has this plus here, and that means it is positively charged. So what happens is that this NAD plus goes towards this glucose, and this glucose has a lot of hydrogen attached to its ends, and it's going to basically rip off one of the electrons within the hydrogen. Now what you then get is a hydrogen that becomes positively charged, and then the NAD that is made is going to rip off that H plus ion, aka proton, and create NADH plus. And to neutralize this new positively charged region, it's going to go to another hydrogen or another part of glucose and rip off another electron to make NADH. NADH. And that is why it's called an electron acceptor, because this is holding electrons. It is going through a process called reduction. Reduction is when you add electrons to yourself. You may hear that word. Oxidation is when you are removing electrons from an atom, or from a, not an atom, but rather a molecule. So in this case, glucose is being oxidized and NAD plus is being reduced. Okay? So, with that being said, recognize the three things that are made and what happens next. So, what happens next is we get this pyruvate molecule and we go to this process called pyruvate oxidation. Remember, oxidation is that you're taking electrons away from the molecule and you're going to be reducing, adding electrons to another molecule. And that is exactly what is going to happen in pyruvate oxidation. See, pyruvate oxidation happens within this region that is not the cytoplasm anymore. This is where we actually get into the mitochondria. Pyruvate oxidation happens within the inner membrane or sometimes within the matrix. And this is an AP test question. I am 90% sure that this was on previous AP tests, so it is your best intention, or it would be in your best interest to figure out this portion of pyruvate oxidation, simply because you're going to get tested on it. So it's best to understand what is made from this entire process. So what happens in pyruvate oxidation? Pyruvate is gone from the cytoplasm, goes into the inner membrane space, and makes the following two things. It makes, and this is going to be, you don't really have to know it too much, but it makes CO2, and it makes the most important thing, acetyl-CoA. Now this is in only one round of pyruvate oxidation. Remember that I said there was 2 pyruvate made. 2. So your net total is going to be 2 CO2 and 2 acetyl-CoA. Okay? That is where you get CO2 in your overall reaction. This is one way you get CO2, but the next is with the Kreb cycle, or where acetyl-CoA goes into next. So acetyl-CoA then moves down from the inner membrane, or even if it's in the matrix, it goes into another portion of the matrix, and it basically starts this process called the citric acid, or the Kreb cycle. And the citric acid cycle is called the way it is because it makes citric acid. It's not something crazy. But I'm going to show you why you don't really have to memorize a lot of this stuff, because look at how complex this is. For AP Bio, you do not need to know this. But what you do need to know is the significance of the citric acid cycle. And the significance of the citric acid cycle is the following. It makes the most amount of electron acceptors. That is why it's significant. So, for example, it makes 6 NADH in 2 rounds. In 2 rounds, remember, you get 2 acetyl-CoA, So it happens 2 times, this citric acid cycle, but you get in total, the net, you get 6 NADH, you get 2 ATP, net total, and lastly you get 2 FADH2. FADH2 is literally the same thing as NADH, but it's a weird thing to kind of describe. It does the same thing. It's just a different molecule essentially. Um, so you don't really have to really worry about the difference between these two things in the AP test. Just note that their initial goal is to do the same thing, and really note that in the citric acid cycle, that NADH and FADH2 are the most important things that are made. But lastly, you also get 4CO2. Okay, so that is the entire process of all of this. Okay, in the citric acid cycle. Um, and remember, another point that is pretty important to make is that this happens 2 times. So in reality, one round of all this only makes half the amount that I have present in front of you. Okay, so that is that. Next up, we're going to go to the most important part. And by the way, this is all happening in the matrix. Sorry. This is happening in the matrix because acetyl CoA goes into the matrix. The next part is oxidative phosphorylation. This is super duper important. I mean, like, if you have to know one part of cellular respiration, you have to know this part. So recall that, and I'm going to take a screenshot of this. Recall that what you have from the citric acid cycle is the NADH. Now, while you are making ATP in the previous 2 out of the 3 cycles, not cycles, but rather processes, you make some ATP. That ATP is negligible to the amount that you're going to make in oxidative phosphorylation. Oxidative phosphorylation makes the most amount of ATP, okay? Now, how does it do it? Well, I'm going to explain it right now. Basically, what happens is the NADH that's made from glycolysis or that is made from the citric acid cycle comes over to the cristae. The cristae. So this white region that I'm now highlighting, this white region is going to be what we're going to be concerned with. This cristae, it is filled with a bunch of important and vital proteins that we have lined up around the lipid bilayer that is there. And what essentially happens is this NADH is going to come up to this protein. It's called cytochrome B, and there are like 1, 2, 3, 4, 5, 6, 7 of these proteins, and it's going to shoot into it an electron. That is why it's called an electron transport chain. I'll explain why it's a transport chain to begin with as well. But that's where the electron part comes from. It shoots into it an electron. Now what you then get is an NADH plus, right? The hydrogen of the molecule loses its electron. And what's significant is as this electron moves into proton or protein 1, the second and third and fourth and fifth and sixth and seventh proteins are all increasingly electronegative. Okay, what that basically means is that their affinity for the electron in their previous protein is much higher. So that means that protein 2 will be more or will have a greater affinity for that electron. It really wants to pull that electron more into itself than what proton or protein 1 wants. And I'm mixing up proton and protein, by the way, I'm sorry, but more than what protein 1 wants. So protein 3 will have a greater affinity for the electron than protein 2 has. 4 has a greater affinity for 3, so on and so forth. So as electron 1 is shot into protein 1 and then begins to move into protein 2, this NADH plus shoots out an H plus ion and becomes NAD plus, and this is all like or not NAD plus, my apologies, just NAD, and it is going to shoot in another electron into now the opened up protein 1. So then you get NAD plus because the first electron is in protein 2, and now the next new electron is in protein 1. And now you create NAD plus, and this NAD plus will essentially go right back into either the matrix or the cytoplasm, and it is going to now continue doing glycolysis and is going to continue doing the citric acid cycle. Now, what's really significant about the reason, oh my god, my apologies, the reason why this is all significant is for the following reason. And as the electrons, and you can see this like dashed line basically, as these electrons go into the next and the next and the next protein, what essentially happens is that these proteins will now shoot up the H plus ions that were created from the NADH, or that were released from the NADH. So what basically happens is that this protein right here, let's say protein 1 and you have protein 2 here. You have the electron sitting right here. Protein 2 really wants this electron, but it's not going to naturally just be able to move that electron over. What happens is that this electron and this protein are actually going to shift. They're going to move around to allow the electron to be in closer contact with the second protein in the electron transport chain, or it could be the third, fourth, fifth, so on and so forth. So, for example, this is the sixth protein, and if there was an electron here, this protein would move over to allow the to allow the electron to end up here, so it can shoot into the seventh protein, or the seventh cytochrome complex, is what it's called. And what basically happens is the following. As this protein begins to shift and really rotate around, it is going to shoot up the H plus ions into its inner membrane space. So this region right here, like between the outer and inner membrane that I was describing about, it's going to start shooting it into there. And what eventually happens is that, let's say the electron transport chain happens enough times, right? Like, let's say I have 2 electrons here, then I have another electrons here, and another 2 electrons here, right? Ignore this one for right now, just simply for the purposes of like the video. So I'm going to have 6 electrons fully occupying this entire transport chain, and that means that NADH has been broken down several times, not only one time, but several times, because there's 6 electrons. So that means that there was 3 NADHs broken down into NAD+. But every single time, remember that they release that H plus into the matrix. What then happens is that as these electrons move down the transport chain and the proteins begin to shift around, they shoot up the protons, or the H plus ions, into the inner membrane. And the reason this is significant is because it creates 2 things. It creates this electrochemical gradient. Okay, and the reason this is significant is that what you essentially are creating is this super positively charged region within the inner membrane space, and a super negatively charged region within the matrix. Okay, because there's less protons in it now, and there's way more protons pumped into the inner membrane space. And also you increase the concentration a lot. Okay, and that's going to be really significant for the following reason. The protons are now a positively charged atom in a very positively charged region. Remember that things that are in the same charge do not like one another. They repel. So as a result of that, the proton is going to be like, okay, I want to get the hell down from here, and I want to go back into the matrix that is more negatively charged. Okay, there's this thing called action potential. Okay, this action potential is created, or this electrochemical gradient. Okay, electro because there is the difference within charges from the matrix and the inner membrane space, and chemical because there is a high concentration of protons. And remember that when things are in high concentration, they always want to naturally go down to low concentration. That's simple diffusion. Okay? Now the problem now is that ions cannot simply diffuse down a phospholipid bilayer. And the reason for that is that the phospholipid bilayer's interior is very hydrophobic because of the tails of the phospholipids being fatty acids. That's unit 2 material. And what essentially ends up happening is that these protons are stuck, but they're highly energetic now. They really want to get down. So what they do is they use this protein called ATP synthase. And this is the most important fellow within our entire story. This is what creates the most amount of ATP, and I'm going to show you a diagram of it. So basically, and this is the best diagram I can really give, the ATP synthase is kind of like a wheel. Okay, and what really happens is that these protons that are really in this positively charged and highly packed space want to quickly move down into this matrix, and they use this motor, this like thing, to sit on. They sit on this motor. So if you can imagine like one protein sits here, then, or not protein, proton sits here, then another one sits here, and another one sits here, and another one sits here, and they begin to push. Because they're so energetic, they push and rotate that entire barrel. And what they do is, after one rotation, a hydrogen proton pushes out. Okay? And what really makes the ATP form is this wheel right here that I'm highlighting or outlining. This wheel right here churns this lower region, and this lower region has a very high affinity for ADP, adenosine diphosphate, and a phosphate group. And that's where all this ADP and phosphates are located. And what this does is, as this motor, or this barrel, this barrel is rotating, and this motor is churning this lower region back and forth in a circular fashion, it is colliding an ADP molecule with a phosphate molecule. It's colliding one another, and it's giving them that kinetic energy that they need to overcome their activation energy and form a molecule, or form ATP. And the reason that so much ATP is made is because there are a lot of ADP and phosphate molecules within this region, because it has a high affinity for these molecules that are, or this part of the protein, right? They really like being in this region of the protein. They really like this part of the matrix, okay? So what happens is that you make 32 ATP. Now, this can range between 30 to 36 depending on, the frequency, and it's totally varies, but just know that it's within that range of molecules. You get that, and you basically create a bunch of ATP now, and you also create a bunch of H plus ions going back into this region. And what eventually happens is that these H plus ions start to float around in the matrix, and now you say, okay, well, what happens to this electron transport chain? Now that I've made my ATP, what else is happening? Well, the electrons are going to find oxygen that floats around within the region. I'm going to erase everything. There is going to be oxygen floating around within this region, and oxygen loves being negative, okay, or negatively charged. So it's going to take in two of the last the last two electrons in this transport chain. Is going to suck them in, and it becomes O2, and it becomes very negative. It becomes a thing called peroxide temporarily. But what's significant is that, remember that there are H plus ions now within this region, and they want to kind of form and become stable. They want to become a regular hydrogen thing. So what happens is that this is a very quick process, by the way, but once the oxygen takes in the two the two electrons, it's going to collide into two other hydrogen molecules that are protons at this point and make water, H2O. That is where water is made in this entire process, okay? So the oxygen takes in the electrons, hits H plus atoms, or hits H plus protons that are coming back from the ATP synthase, and it forms water, and ATP is made as well, okay? So really what you get in this reaction is the following. You have your NADH, and your NADH gets broken down into a proton, and two electrons. Now these electrons and these protons go through their electrochemical gradient, or through the transport chain, and they end up making H2O, and they end up making 30 to 36 ATP. Okay, that's really the entire process. And I guess you also technically make NAD+, which ends up going back into the cytoplasm or the matrix to then continue the citric acid cycle, or to continue glycolysis. Okay, that's basically all that for the electron transport chain, or oxidative phosphorylation. Okay. So I kind of explained what everything is. So I kind of explained 8, 9, what are the high electron carriers involved in cellular respiration. 10 is the NADH, or NAD+, or FADH. And by the way, note that it's not only NADH that does this process of shooting in electrons into the electron transport chain. It could also be FADH too, to make FAD eventually. So that also happens. Which ions are involved in the transport chain? Those are protons. Electrons are also involved in the transport chain as well. What is the final electron acceptor in the electron transport chain? Oxygen. The step works by basically shooting the electrons into the oxygen, then the oxygen finds H plus ions and makes water. What is the name of the enzyme that generates ATP? For cellular respiration, where is the enzyme embedded? The enzyme is embedded on the cristae. It's called ATP synthase. Super, super important. Know it. Now, next up is fermentation, right? It asks what happens when there is no oxygen present. And that is actually kind of a worrisome thing. So basically, imagine the following. I'm going to erase everything. Imagine the following. The oxygen, and I'll give you a perfect example. When you are running a lot, really, really quickly, you are breathing in and you are releasing a lot of oxygen. Or, well, you're, you're releasing CO2, but you're breathing in a lot of oxygen. And you are going to be depleting your storage of oxygen because you're rapidly making up energy. Okay, you're really, really quickly making up energy. And you're going to not have enough oxygen to necessarily be able to create as much ATP. And what essentially happens in this case is that your electron transport chain gets blocked. Because why does it get blocked? Well, the O2 is so much depleted that you're not going to be able to accept electrons. So your electrons are just going to be stuck all within this transport chain. Okay? But the problem is that I'm in my citric acid cycle and my glycolysis, and my glycolysis, I'm rampantly producing NADH and FADH. Okay? I'm rampantly producing FADH2 and NADH, right? And because I'm producing it so much, I expect them to kind of come back and get reused. And they do get reused under normal circumstances in which the oxygen is present and can basically accept the electrons and allow that NAD plus to kind of go back, how I mentioned, into glycolysis. But what happens is that the NAD plus does not become um, a molecule anymore because the electron transport chain is blocked. So the FADH2 and the NADH that is created cannot keep on shooting its electrons and allow this cycle to continue. So you're going to continue to accumulate a lot of NADH and FADH2, and you essentially block glycolysis, or not block, well, you block glycolysis from occurring, and you essentially block the citric acid cycle to occur as well. Because it will no longer have the required um what you call it the required electron acceptors or electron acceptor carriers to continue the cycle to be made. And I cannot make ATP, right? In any case, really, because if I cannot um, undergoes glycolysis, if I cannot undergo citric acid, or if I cannot undergo the citric acid cycle, my apologies, I can't make the 2 ATP that are made in both those cases, but also my electron transport chain is blocked. I can't even make ATP for myself because I can't pump an electrochemical gradient. I can't make that because it's all clogged, right? I need to continuously keep this pushing. So now I'm really worried that my worry now is that I cannot do cellular respiration. In particular, I cannot make a lot of ATP because my transport chain is blocked, right? That's the biggest concern because that's where most of the ATP is made. I cannot make ATP. Can't make ATP. So my body undergoes this thing called lactic acid, and I'll show you the equation. It's not necessarily required, but lactic acid fermentation equation. I will undergo lactic acid fermentation, which basically what I do is the following. I will get glucose, right? And this is going to be in glycolysis. So we're going to go back to glycolysis. And this is how our body supplements ourselves when we're running low on ATP. And this is very temporary, but it's called fermentation. Basically what I do, and this is a form of anaerobic respiration because I'm not using oxygen to um, to create ATP. I'm rather using, other methods. Aerobic aspiration would mean, and these are just two key terms you should know. Aerobic aspiration would mean that you use oxygen. Let me just double check that. I think aerobic aspiration means, yeah, you use oxygen to make your fuel or ATP. Now, basically what happens is this is the equation for it. But essentially, when you're doing or when you're creating glucose, or when you're using glucose, you make your 2-pyruvate. You make your 2-pyruvate. You make your 2 ATP, which is going to be the important part, and you make your 2 to NADH. Now what happens next is the following. This is the very, very important part. You make pyruvate into lactate, okay? You make it into lactate. And the reason this is significant, and this is probably a better form of what I'm showing you, is that as you are making pyruvate into lactate, you actually require NADH to give up a few electrons within the process and become NAD plus again. And once you create NAD plus, you can redo glycolysis again to create more ATP for yourself. And this is happening really, really quickly. But again, you're going to strain your body and eventually lose supplies. But for a temporary amount of time, this is how you are going to supplement your body, that you get the pyruvate and then you end up actually breaking down the pyruvate into lactate, and when you are making lactate you use your NADH to essentially give lactate electrons and release a proton in the process to again make NAD+, and that NAD+ can then be reused into glycolysis. That is fermentation. That is the fermentation that occurs in our body. There's another type of fermentation, but that is probably not going to be asked of you on the actual test, so I wouldn't worry if I were you. So next up, what is the balanced equation of cellular respiration? C6H12O6 plus O2 makes ATP plus CO2 plus H2O. Remember, H2O is made in what? Oxidative phosphorylation. CO2 is made in the Kreb cycle, pyruvate oxidation. And these two are the things that you start off with. Oxygen is really used as the final electron acceptor. ATP is made in 3 out of the 4 stages of cellular respiration. But the balanced equation of all this, basically when I want to match all the total amount of molecules with one another, so I want to ensure that the same amount of carbon is within both sides, and the same amount of oxygen is in both sides, is going to be the following values right here. I believe it is, you get, no, not this, sorry, 6 CO2 with 6 O2 and 6 H2O. Okay, so C6H12O6 plus 6O2 plus ATP, or forms ATP plus 6CO2 plus 6H2O. That is the balanced equation. Remember that equation, by the way, but also remember the following, that not only is this made within the reaction, but also heat is generated. Now, if you want, and this is super important because I recall a question on my AP test that asked about heat formation during this entire process. Heat is formated during all the reactions that happen, okay? That is through entropy. Entropy is increasing, so as the reactions take place, heat is released. And by the way, this is catabolic, so of course heat is released. And the majority, by the way, of the energy used, or the majority of everything that happens, is released as heat. I think this is about 40% only, and the rest is like, its own percentage, but heat takes up the majority of things that are released. And a lot of animals, and this is just a kind of a nuanced thing that happens, is that a lot of animals actually do not want to, during hibernation, make a lot of energy. They kind of want to hibernate. So what they do is they have another protein. Instead of ATP synthase, they have another protein to harness all the energy that is coming down from that electrochemical gradient that is formed here in the in the inner membrane. What they essentially use is, instead of ATP synthase, a thing called a decoupling protein, where they essentially push down, or allow all the protons to push down. The electron transport chain is the same, by the way, but they allow all the protons to push down and release their energy as they go through that motor, this motor. But as they do that, instead of creating ATP because they don't need energy, they create heat through that process, allowing them to kind of stay heated during a period of very cold time. That's kind of what bears do. That's called, and this is typically in fat, brown tissue. Okay, so be familiar with that. It's a thing that is not always going to pop up on an AP test or an AP question on your unit test, but it's still a possibility, and you kind of want to be able to get as many points as possible. This definitely the difference between an A and a B on a test, maybe. So just recognize that. But that, in reality, or in its totality, is all of cellular respiration and fermentation. So that being said, let's kind of begin the next process, and that is photosynthesis. And photosynthesis is not that tricky. It's almost the same process, but basically in reverse. So very generally, photosynthesis captures energy from the sun and produces sugar. Okay, but you have to go way more specific to understand this. What kind of cells undergo photosynthesis? Typically, eukaryotic cells undergo photosynthesis, although there are some bacteria that can do that as well. But for the premises of this chapter, or really this unit, know that it's always typically plants. This is how they make their own food. But also, you have a lot of algae that can also do photosynthesis too, and those are eukaryotic cells, but they're a different type. Anyways, circle one, photosynthesis first evolved in what? Prokaryotic, eukaryotic organisms. Prokaryote, remember that photosynthesis comes from the chloroplast, and chloroplast follow the endosymbiote theory as well. Okay, where does the photosynthesis process take place? So it's really in the chloroplast, but there are two parts of the chloroplast. There is the vacuoles and this stroma, okay, and of course the outer and inner membrane. And if I were you, I wouldn't really be concerned with that. I would be more concerned with the stroma, this region right here. This is the jelly-like fluid. It's like a cytoplasm within the interior, and the vacuoles, which are these like stacks, these like chips of everything. A stack of this is called a granum. One individual chip is called a thaculoid. Okay, or am I pronouncing that right? Yeah, thylakoid. Whatever. What are the different parts of this organelle? I just explained it, the ones that you have to know. What are the steps of photosynthesis? It is a two-step process. There are light-dependent reactions, light-dependent reactions, light-dependent reactions, and then the Calvin cycle. Now, for the purpose of the AP test, this is the part that is super duper important. The Calvin cycle is, I'll be honest with you, it is they do not care about it on the AP test, even though that is literally where most of the sugar is made, and it's technically not sugar. I'll explain that in a second. But really, what happens during the light-dependent reaction system? So the light-dependent reactions happen on the thylakoids or the vacuoles, okay? And the vacuoles basically have their own membrane. And what really happens here is the following. There is a protein, or it's not a protein, but rather a complex of molecules called photosystem 2, okay? Photosystem 2. And really what's inside of all these regions are different colored things. It's a really poor description of it, I know, but there are, God, I forgot the specific word. I'm not sure if your thing has it here. No, it doesn't. Well, basically what's here is just chlorophyll, and chlorophyll is this molecule that can absorb energy, and what you essentially do, and this is a little bit of chemistry, by the way, but as light shines into the thylakoids or vacuoles of the chloroplast, what happens is that it excites electrons. And as the electrons get excited, right? So let me show you what chlorophyll looks like. Okay. Um, Molecule. Okay. This is the chlorophyll molecule. This is the important thing. So light will hit this molecule, and you do not have to know this molecule, but I'm just showing it to you guys for just the purpose of the video. As it hits this molecule, let's say it hits a region right here, and when it hits it, what happens is that it'll excite an electron. Basically, it'll shoot it up into a higher energy state. This is a lot of chemistry, by the way, so you don't really have to know it, but it excites it. And as this electron shoots up because it's got an energy, it'll come back down and release the energy of the light, and it'll kind of zigzag around within the photosystem. If you see these green things, that's basically what's happening. Um, let me make this bigger. This right here is going to get shot in with light. This is going to excite an electron. The electron is going to go back down, or it'll shoot up, like basically shoot an electron out of its orbit, and the electron then, because it becomes really unstable, wants to come back down, so it releases energy, shoots it back down here, and goes into the next form of chlorophyll, and then it goes into the next one as the process continues, and it goes and goes and goes, until it reaches one specific region of the photosystem 2. And that's this molecule right here, this Mg. This is chlorophyll A. Okay, and it'll go to this magnesium atom, and what happens is the electron that got excited before it, right? So this one right here, will shoot the energy that it has into this magnesium atom, and that magnesium atom is going to shoot out a literal electron out of it, and it is going to go into the electron transport chain, much that is basically similar to how it works in the how it works into the what you call it, the the uh, cellular respiration process. Okay. And really the way this all works is the following. As you see, the electron gets shot out into it and goes into the electron transport chain, and the electron transport chain, ironically enough, is going to do the following. It is going to bring in protons from the chloroplast stroma into the region of the thalloid lumen, which is like the inner region of the chloroplast. If I have a photo, I'll show you this right here, chloroplast diagram. Okay. The lumen is this very light green region of the thalloid. So this is the stroma, the thalloid lumen is here, and this is the region where actually like this outline right here, where my like this darker green portion right here, that is where this process is taking place. But essentially what happens is the following. The proton will again kind of like how in cellular respiration the proton gets shot into the matrix. In this case, the proton gets shot from the stroma into the thalloid lumen, and what happens is that the electron will now go through this transport chain, and it'll basically wait in this last proton, or not proton, but protein, my apologies again, protein, right here, plastocyanine, okay, and light is going to hit photosystem 1 now, okay, at a different, variation of light, a different wavelength will hit it. This favors light on the 700th wavelength, while photosystem 2 favors on the 680th wavelength, or the values of, or the length of the wavelength. You don't need to know it. But that's basically what it is. And what that does, it excites another electron, and this electron will then go through another system of of electron transport, but this one instead does not release protons into the thalloid lumen. It's just simply going to allow the electron to continue going through and going through, and then it reaches this protein called NADP reductase. And what NADP reductase does is that it gives NADP, which is the electron carrier acceptor in this case, it gives it the electron, and then it becomes NADP minus, and then it brings in a proton from the stroma, and it will then create NADPH. That is going to be super duper important for the Calvin cycle, okay? Kind of like how NADH is used in oxidative phosphorylation. NADPH is going to be used for the Calvin cycle, so those are kind of like synonymous for one another. They kind of act in the same way. So what ends up happening now is that, okay, you have your NADPH, but you have an issue. You have an electron missing within the magnesium here, and you have an electron or the magnesium from the chlorophyll, right? And you have an electron missing within photosystem 1, because really it's kind of the same structure, right? It looks like this. You're missing it. And where do you get these electrons? Well, you actually get them through the following. You end up breaking water. There is a enzyme at the bottom of photosystem 2, right, like located where my cursor is, and actually for the purpose of this I will screenshot this. There is an enzyme here that ends up actually breaking water into oxygen, right? This is where you get your oxygen out of this equation, and protons. And what ends up happening is that you create, and this is your lumen, and this is your stroma, right? So this is kind of like the gooey part, like if you remember how this is this region right here, and the lumen is the interior of the thalloids. So what you end up getting is a very high concentration of H+. You create an electrochemical gradient. Remember, why is this electrochemical gradient being created in the lumen? Well, you have a lot of H+ floating around because H2O got broken into oxygen and protons, and the electron transport chain is shooting in H+ atoms into the lumen as well, similar to how the matrix is shooting H plus atoms into the inner membrane. It's kind of like the opposite in this situation. Anyways, you create a very positively charged lumen. Lumen is positively charged, and this region is going to become negatively charged, the stroma. And much like how H plus wants to basically kind of go down this electrochemical gradient, the same thing is going to be happening in this case as well. And the H plus within the lumen wants to kind of go into the stroma because it hates being positively charged. It hates the high concentration of it all. So what ends up happening is that it goes into this ATP synthase, it turns the wheel, and it forms a bunch of ATP. Now, in this case, the ATP created in the light-dependent reactions, by the way, is not going to be the ATP used for cellular respiration. It's going to actually typically be used within the Calvin cycle, okay? So that's basically the entire light-dependent reaction. Just note that it's really in the thaculoid. You're going to have to deal with the stroma, the lumen, so on and so forth. So that's that. Next up is the Calvin cycle. Calvin cycle is a little bit different. So this is going to happen in the stroma, okay? So the stroma is this region right here, okay? So because now I have, brought in my NADPH, right? Because remember my light dependent reaction, right? This is the stroma, right? Oop. I'm going to screenshot this. This is a kind of good photo. This is my stroma, and I have shot my NADPH in here. I have my ATP for the Calvin cycle that is required in here as well. And I am shooting it in to kind of create the following. So what basically happens is a really complex process. You don't really need to know much of it, but you basically have this product called Rubisco, and carbon fixation is a process that is the first step of the Calvin cycle. And what you're essentially doing is you're taking carbon, CO2, from the outside air, okay, that is taken in by this part of the cell or of the plant called the stomata. The stomata is like these cracks of a plant leaf that are open that allow CO2 to seep in. This is where you're going to get CO2, okay? You're going to get the NADPH that you have, and you're going to get a lot of ATP, and you're going to create the real sugar out of all this, okay? This is where the sugar is made, the Calvin cycle. And the reason that you had the light-dependent reaction is only to basically make the NADPH and to make extra ATP. And to make oxygen as well. But now this is where you get carbon from outside through the stomata of the leaves, and you're going to essentially fix it, basically make it from a gaseous form into a solid or a solidified form, okay? And that's basically what you're doing. You get this molecule called Rubisco. Rubisco, I'll show you it. Structure. It's really big, super huge, but all you really have to know is that you see all these carbons in Rubisco. Well, what you essentially do is you break Rubisco into two molecules that are smaller than one another, okay? And once you break Rubisco up, what you end up doing is you can create sugar, okay? But it's not your conventional sugar, and this is in the reduction stage, so this is where you're adding electrons to the broken apart Rubisco molecule, which basically looks, actually, you know what, let me screenshot this. Rubisco is one huge molecule, okay? You get 3 molecules of CO2, you add it to Rubisco, you make 6 molecules of 3-PGA, which you don't really have to know, just think of it as an intermediate. And what essentially you do is you add 6 ATP, you add electrons to these molecules, and you can end up making it into the thing called G3P. And G3P is sugar. Now, is it your conventional sugar? No, it is not C6H12O6. It is CHOH with CH2OP. That's a phosphate group. That is the sugar that you make. Now, this ends up eventually becoming this eventually ends up becoming glucose through another process, but that is not covered in the AP Bio curriculum, so you don't have to worry about it. But just know that G3P is made. So again, you get Rubisco, you add CO2 to it, you make it a very unstable molecule, which then breaks, and this happens like several times. You do it to several Rubiscos. You do it to 3 because you have 3 CO2 molecules. You make 6 molecules of a compound called 3-PGA. The reason it's called 3 is because it's 3 carbons. And what ends up happening is the following: you have 3 of these, or you have 6 3-carbon molecules, okay? And in total, only 2 of them are going to end up actually becoming one of them, I believe, is going to end up actually becoming a molecule that is used for sugar, like actual sugar that the plant uses to eat. And the 5 other molecules of 3-PGA are actually used to create Rubisco again, to recycle this process. And remember, this process happens 2 times. 2 times. So in total, you're going to be creating 2 uh, partial glucose molecules. Let's just call that partial glucose molecules. In reality, it's G3P. You're going to create 3 of them. And the reason that you're going to be creating 3 of, or only 2 of them in total, is because the cycle happens 2 times, and each time you create 6 PGA molecules right here. And out of those 6 PGA molecules, as you reduce them, as you add the NADPH and you add the ATPs to it, you're only going to be able to use 1 out of the 6 molecules as a form of sugar, and the 5 other become Rubisco eventually through a process called regeneration. And after they regenerate, the cycle happens another time, okay, with another 3 molecules of CO2, creating another 1 molecule of G3P, which is in total 2, okay? That's really the Calvin cycle. Not really needed for, not really needed for the AP test again, but just kind of you have to know it. But the biggest point is that real sugar is not made in the Calvin cycle, okay? And the overall equation of this is basically the same one as cellular respiration, but for photosynthesis you have the following. You're going to originally have H2, H2O plus CO2. You're going to make C6H12O6 plus O2 plus, I guess you can say ATP in a way, but I wouldn't necessarily like say you should include that, because the real thing that most AP test graders are looking for is the fact that you're able to recognize that sugar is made and oxygen is made within this process, and also never forget to balance it. And the balanced equation for all this is going to become 6H2O plus 6CO2 forms C6H12 H12O6 plus 6O2. Question is, where does the O2 come from? The O2 comes from the breakage of water. Remember that H plus is made, and the electrons, oh, I forgot to mention this, electrons from the H plus are shot back into the magnesium. I think I talked about this, but yeah, the electrons, 2 of them are shot back in, so both of these can kind of be satisfied. Hope that was clarified if that was confusing for anyone. But that's that. I also want to know one more thing, that um, in cellular respiration, and this is sometimes um, a thing that isn't really talked about, but in cellular respiration, is sugar the only molecule used to create ATP? No, fats or fatty acids can be used. Different sugars besides glucose can be used. But what ends up happening really is that amino acids, and by the way, amino acids can be used as well. Amino acids, fatty lipids, and other sugars can be used in cellular respiration as a replacement for glucose. However, what ends up really happening is that they eventually become some way, somehow, into pyruvate or acetyl-CoA. So they're just used as things called intermediates. You may be asked that on your AP test. You may be asked on your unit test. Who knows? Just be aware of that. But without much else, I believe this is the end of your video. So best of luck, everyone, to whoever's going to be taking the AP test, and if you're watching this video, hopefully it helped. Anyways, good luck.